Every single morning, without thinking about it, you do something that women in ancient Egypt were doing 4,000 years ago. You frame your eyes, you extend your lashes, you reach for a brush or a pencil or a curler, and you try to recreate, artificially, temporarily, imperfectly, something that a tiny fraction of women on this earth are simply born with. And here is what nobody tells you. Those women didn't earn it. They didn't work for it. They didn't choose it. A gene inside their body, a single gene, sitting on chromosome 4, made a mistake. And that mistake produced the most coveted physical feature in the history of human beauty. Researchers have a clinical name for it. They call it trichomegaly. They classify it as a disorder. They write about it in medical journals under the category of rare diseases. But outside those journals, in the real world, in every culture that has ever existed on this planet, in ancient Egypt, in Mughal India, in Renaissance Europe, in modern-day Korea and Brazil and New York, women with this condition have been stopped in the street, painted in portraits, written about in poetry, asked over and over again what mascara they use. They don't use any. That's the answer. They never needed to. Now here's the question that scientists spent decades trying to answer and finally cracked. Why do some people have eyelashes that grow twice as long as everyone else's? What is happening inside their bodies that doesn't happen inside yours? And why does the human brain respond to long lashes the same way? In every culture, in every language, in every era of recorded history, before a single word is spoken, before a name is known, before any conscious decision is made. The answer is in a gene most people have never heard of. And once you understand what it does, not just to eyelashes, but to cats, to sheep, to Angora rabbits, to the entire mammalian kingdom, you will never think about a pair of eyes the same way again. Every eyelash on every human face goes through a continuous cycle of 3 phases. The first phase is called anagen. This is the growth phase. During anagen, the hair follicle at the base of each lash is alive, deep, and metabolically furious, fed by a rich blood supply that delivers oxygen, amino acids, and the molecular signals that drive the cells in the follicle matrix to divide rapidly and continuously, pushing a new lash upward through the skin. About 30 to 40% of all eyelashes are in the anagen phase at any given time. This phase lasts about 30 to 45 days. 30 to 45 days, that is the crucial number. That narrow window of active growth is the biological reason that eyelashes don't grow to your shoulders. While the hair on your scalp stays in anagen for 2 to 7 years, long enough to reach your waist if you let it, your eyelashes stop after about 6 weeks. Something shuts the growth phase down. Something sends a signal to the follicle that says, that is enough. Stop here. The second phase is catagen. The follicle detaches from its blood supply. The lash stops growing. The follicle begins to shrink. And then the third phase, telogen, the resting phase, begins, during which the lash simply sits in the follicle, no longer growing, until a new lash forming beneath it pushes it out. The telogen phase can last up to 100 days, depending on the individual's lash cycle. The entire architecture of eyelash length. The reason they frame the eye rather than droop across it comes down to one biological fact. The anagen phase for eyelashes is genetically programmed to be short, precisely short, calibrated short. And the molecule responsible for delivering the stop signal, the molecule that tells the actively growing follicle that its time is up, is the protein produced by the FGF5 gene. FGF5 functions as a negative regulator of the hair cycle in mammals. It is expressed in the outer root sheath of hair follicles during the late anagen phase of the hair cycle. It functions as a signaling molecule, mediating the transition of the anagen growth phase to catagen regression phase of the hair cycle. FGF5 is, in the most fundamental sense, the brake on hair growth. It is expressed precisely at the end of the growth phase, released into the outer root sheath of the follicle like a chemical command, and it triggers the cascade of events that ends anagen and begins the retreat. Without FGF5, the follicle doesn't receive the stop signal. The anagen phase simply continues, and the lash keeps growing. Here is where the story becomes extraordinary. In 2014, a team of researchers at the University of Manchester and the Wellcome Trust Sanger Institute were studying families from Pakistan in which multiple members had the same unusual trait. Eyelashes so dramatically long that they caused corneal irritation, brushed against eyeglasses, and required regular trimming. The condition had a name, trichomegaly, from the Greek for hair and large. It had been documented in the medical literature since 1944, when a physician named H. Gray first described it and suggested the alternative name movie lashes, referring to the then fashionable image of glamorous Hollywood actresses. But nobody knew what caused it. Nobody knew what gene, what mutation, what biological mechanism produced eyelashes that grew far beyond the normal limit and refused to stop. Mechanisms that regulate the growth of eyelashes had remained obscure. That sentence appears 3 times in the PNAS paper, not for emphasis, but because the researchers wanted to be clear about what they had inherited from the previous century of science, a complete mystery. Then they sequenced the genomes of the affected family members. Using a combination of whole exome sequencing and homozygosity mapping, they identified distinct pathogenic mutations within fibroblast growth factor 5, FGF5, that underlie the disorder. Subsequent sequencing of this gene in several additional trichomegaly families identified an additional mutation in FGF5. Every affected family member had a mutation in FGF5. Every unaffected family member did not. The mutation didn't create something new, it destroyed something old. It broke the FGF5 protein, the stop signal, so that it could no longer function. And without the stop signal, the anagen phase of the eyelash follicle simply continued. The growth phase that was supposed to last 30 to 45 days ran on and on and on, producing eyelashes that grew far beyond any normal biological limit. Hair fibers from forearms of these patients were significantly longer than hairs from control individuals, with an increased proportion in the growth phase anagen. It wasn't just the eyelashes. Every hair on the affected individuals' bodies was growing longer than normal, because FGF5 regulates the stop signal across all follicles, not just those on the eyelids. The eyelashes were simply the most visible manifestation of a systemic change in how the body manages the hair growth cycle. And then the researchers did something even more revealing. They used hair follicle organ cultures, laboratory models of living follicles maintained outside the body, and demonstrated directly that FGF5 induces regression of the human hair follicle. When FGF5 was present, follicles retreated from anagen. When FGF5 was absent or non-functional, they stayed in anagen longer. The mechanism was not theoretical. It was observed, measured, and replicated. The break was real. And breaking it produced exactly what the affected families already knew from experience: extraordinary lashes that the world found beautiful and that doctors classified as a medical condition. But this story doesn't begin with humans. It doesn't even begin with mammals alive today. Subsequent to identification of FGF5 as causative for the Angora mouse phenotype, genetic variants in FGF5 have been shown to underlie hair length regulation in several other species, including cats, dogs, and even rabbits. The Angora mouse, the Angora cat, the Angora rabbit. Those luxuriously long, silky-coated breeds that people have kept and marveled at for centuries. All of them carry mutations in FGF5. All of them are running the same biological experiment as the Pakistani families, with trichomegaly, a broken stop signal, a prolonged anagen phase, hair that keeps growing because the molecular command to stop never arrives. Mutations in the FGF5 gene result in longer hair in mice, donkeys, dogs, cats, goats, sheep, donkeys, llamas, dromedaries, rabbits, and humans. The same gene, the same mechanism, the same outcome, longer hair, across species separated by tens of millions of years of evolution. That is not coincidence. That is the most fundamental evidence of evolutionary conservation, a biological mechanism so important, so precisely calibrated, so essential to the proper functioning of the body, that it has been maintained nearly intact across the entire mammalian lineage. When you look at an Angora cat and find it beautiful, when you reach out to touch that impossibly soft, impossibly long coat, you are responding to the same broken gene that produces human trichomegaly, the same mutation, the same prolonged anagen, the same follicle that never received the stop command. The cat breeders who selected for longer and longer coats across generations of Angoras were, without knowing it, selecting for FGF5 dysfunction. They were breeding a more and more broken break and calling the result beautiful. And now, knowing all of this, look at what happens when a woman applies mascara. Look at what she's doing with her hands and her mirror and her small brush. She is artificially extending the visual signal of long lashes. She is mimicking, with pigment and product, what a broken FGF5 gene produces naturally. She is chasing the appearance of something that nature only gives to people with a specific mutation in a gene on chromosome 4. And she is doing it because something in her visual system, and in the visual systems of the people who will look at her, responds to that signal with an unmistakable response that centuries of evolutionary psychology have now begun to understand. What does that response feel like from the outside? What actually happens in the brain of the person who looks at a face with long lashes? The science on this is precise, and it is surprising in ways that the beauty industry has not yet caught up to. In 2020, Dr. Farid Pajouhi of the University of British Columbia led a study that, for the first time, systematically tested the relationship between eyelash length and perceived attractiveness. They used 3D rendering software to generate female and male faces with eyelash lengths ranging from none at all to half the width of the eye opening. They showed these faces to hundreds of participants and asked them to rate attractiveness. The result destroyed the assumption that longer is always better. Results showed that both health and attractiveness perceptions follow an inverted U pattern, peaking at eyelash lengths about 1/3 the width of the eye, consistently across different ethnicities and regardless of the participant's sex. An inverted U. Not a slope, not a curve that keeps rising. A peak and then a fall. Eyelash attractiveness rises as length increases, up to a point. And then, past that point, it falls again. Eyelashes that are too long are rated just as unattractive as eyelashes that are too short. Both very long and very short eyelashes can signal disorder or disease. And what is the peak? What is the exact length the human brain finds most attractive? The optimal eyelash length for women is about 1/4 to 1/3 of the width of one's eye. The optimal eyelash length for men was about 1/5 of the width of one's eye, 1/3 of the eye width. That number will come back in a moment because it appears in a completely different line of research, from a completely different discipline, reached by a completely different method, and its reappearance is one of the most elegant convergences in the recent history of biological science. But first, consider what that finding means for every mascara advertisement ever made. Every image of impossibly long, impossibly thick lashes on a magazine page or a phone screen, those lashes are often longer than the optimal 1/3 ratio. Which means the images the beauty industry uses to sell the ideal of long lashes frequently depict lashes that the human brain, by its own evolved preferences, would find slightly less attractive than shorter alternatives. The aspiration and the optimum don't align. The industry is selling beyond the peak. Now, to the other side of this, the aerodynamic side. The science that changed the way biologists think about why eyelashes exist at all. For most of human history, the assumption was simple. Eyelashes are a filter. They catch debris. They keep dust and insects out of the eye. They are nature's net, strung across the eye opening, catching whatever the wind blows. That explanation seemed obvious and complete. Nobody questioned it seriously until 2015, when researchers at the Georgia Institute of Technology did something nobody had thought to do before. They measured eyelashes. Not just human eyelashes from 22 different mammalian species, including elephants, hedgehogs, camels, and humans. And they put eyelids with and without lashes into a wind tunnel. A landmark 2015 study published in the Journal of the Royal Society Interface found that eyelash length across 22 mammal species is consistently 1/3 the width of the eye opening, an optimal ratio that appeared to have evolved independently across the mammalian lineage. 1/3. The same ratio, the one that appears in the attractiveness research. The one the human brain finds most beautiful is the one that 22 different mammalian lineages, through millions of years of independent evolution, converged on as the optimal protective length. At this specific length, eyelashes create a boundary layer of still air just above the ocular surface. Not a filter, not a net, a buffer zone, a region of reduced airflow directly above the cornea and tear film, where air velocity drops low enough that particles carried by the surrounding air can no longer reach the eye surface. The lashes don't catch debris. They deflect the airflow that carries debris. The architecture is aerodynamic, not mechanical. Eyelashes divert airflow to protect the eye, reducing both dust landing on the eye and moisture evaporating from the tear film by about 50%. 50% reduction in tear film evaporation. That is a number with enormous clinical significance. Dry eye syndrome, the burning, the redness, the visual disturbance that affects hundreds of millions of people globally, is driven by excessive evaporation of the tear film. And healthy, properly proportioned eyelashes reduce that evaporation by half, not through any active mechanism, not through any biological process, simply by being the right length and creating a still air boundary layer above the eye. When eyelashes are too long, they extend beyond the boundary layer. They curve down and touch the cheek or the nose. They direct airflow toward the eye rather than away from it. Longer lashes are associated with more evaporation. Therefore, dry eye risks are higher. The eyelash that grows past the optimum, the one that looks dramatic, that women spend billions of dollars trying to replicate, is, from a purely biological standpoint, worse at protecting the eye than shorter lashes. It is beautiful to the human brain and harmful to the eye at the same time. Does that tell you something about the distance between what evolution optimizes for and what culture aspires to? The brain was calibrated by millions of years of selection to find the 1/3 ratio most attractive, because that ratio corresponds to health, to proper function, to an eye that is well protected and well maintained. And then culture took that preference and tried to amplify it, to extend it, to push it further, and in doing so, created an aesthetic standard that is simultaneously appealing to the eye that sees it and harmful to the eye that bears it. Now, return to FGF5. Because the discovery of this gene as the regulator of eyelash length didn't just explain trichomegaly, It opened a door that the cosmetic industry walked through almost immediately. The researchers uncovered a therapeutic target to selectively regulate eyelash growth. If FGF5 is the stop signal, then partially inhibiting FGF5, reducing its activity without eliminating it entirely, would extend the anagen phase of eyelash follicles, producing lashes that are longer than they would otherwise be. Not as long as full trichomegaly, not so long they cause corneal irritation, but longer, thicker, more visible. This is not theoretical. The connection between FGF5 and eyelash growth has already been validated in the drug research literature by a different route. Trichomegaly of eyelash is defined as excessive increase in the length, thickness, stiffness, curling, or pigmentation of eyelashes. Acquired forms of trichomegaly were reported in certain drugs including zidovudin, latanoprost, bimatoprost, cyclosporine, topiramate, tacrolimus, and interferon alpha, bimatoprost, latanoprost. These are prostaglandin analogs, drugs originally developed to treat glaucoma by reducing pressure inside the eye. In the early 2000s, ophthalmologists noticed something unexpected happening to their patients. Eyelashes were growing longer, not dramatically long, not pathologically long, just noticeably, unexpectedly, remarkably longer. The patients were not complaining. They were asking how to keep the effect. The mechanism of action of these prostaglandin drugs intersects with the same hair follicle signaling pathways that FGF5 regulates, specifically the duration and depth of the anagen phase. By modifying prostaglandin receptor signaling in the follicle, these drugs extended the growth phase of eyelash hairs in ways that FGF5 dysfunction achieves genetically. The FDA ultimately approved bimatoprost for eyelash growth in 2008 under the brand name Latisse, the first and so far only drug approved specifically to grow longer eyelashes. What Latisse does with chemistry, the FGF5 mutation does with genetics. What the genetics does naturally in the families from Pakistan, the drug does temporarily in every person who applies it. 3 completely different routes to the same destination. An eyelash anagen phase that runs longer than the body intended, producing lashes that cross the line from ordinary to extraordinary. And extraordinary they are, not because science says so, because 83 genes in the human genome are currently associated with the phenotype of long eyelashes. 83 genes whose variants influence whether a given person will develop longer than average lashes. FGF5 is the master regulator, the one gene whose complete loss of function produces the most dramatic outcome. But it operates in a genetic environment that includes dozens of other genes influencing follicle depth, keratin production, anagen sensitivity to growth factors, and the precise cellular architecture of the eyelid. Long eyelashes, in most people, are not the product of a single broken gene. They are the cumulative output of a network of variants, each contributing a small amount to a trait that the brain then processes as a coherent visual whole. The average human has 75 to 80 lashes on the bottom eyelid and 90 to 160 lashes on the top eyelid. Lashes grow at 0.12 to 0.14 millimeters per day until they reach their optimum length, usually 12 millimeters or less, then fall out on their own. Millimeters, that is the average natural limit. A number determined not by any arbitrary biological accident, but by the interaction of FGF5 activity with the specific follicle characteristics of the eyelid, the depth of the follicle, the intensity of its blood supply during anagen, the sensitivity of its cells to the FGF5 stop signal. Everything about the 12 millimeter length is the output of a system so precisely regulated that it has been conserved, in its essential architecture, across every mammal alive today. And trichomegaly, familial trichomegaly, the genetic form caused by FGF5 mutation, is defined medically as eyelashes measuring 12 millimeters or greater in the central area and 8 millimeters or greater in the peripheral area. The very threshold of beauty and unusualness is the exact point where normal FGF5 function ends and FGF5 dysfunction begins. The gene's stop signal is precisely calibrated to produce lashes at the upper limit of what the mammalian eye protection system can optimally use. And the mutation that breaks that signal produces lashes that cross into a territory that humans find beautiful and that science classifies as a rare disease. Think for a moment about what a woman does every morning at a mirror. Think about what she is doing, in the most literal biological terms, when she applies mascara or uses an eyelash curler or considers the options that the cosmetic industry has spent a century developing. She's attempting to modify, externally and temporarily, the output of a gene system that was calibrated over millions of years of mammalian evolution. She is trying to move her lashes toward a longer length, not infinitely longer, not movie lash, trichomegaly length, but closer to the upper boundary of what the brain perceives as attractive, which is exactly where natural FGF5 variation places the longest lashes in the normal population. She is not chasing an arbitrary beauty standard. She is chasing a signal that the human brain has been calibrated by evolution to notice. A signal that, across all ethnicities, in all cultures, on every face, assessed in the 2025 attractiveness study, published in Archives of Sexual Behavior, consistently peaks at 1/3 of eye width and falls on either side of that optimum. Results confirmed that both health and attractiveness perceptions follow an inverted U pattern, peaking at eyelash lengths about 1/3 the width of the eye, consistently across different ethnicities, and regardless of the participant's sex. The perception of health, the perception of attractiveness. These are not separate signals processed by different brain systems. They are the same signal, arising from the same visual input, processed by the same neural circuits that evolution built to read the face of another person and extract the information that matters for survival and reproduction. What looks healthy looks attractive. What looks attractive is telling you something real about biological function. Eyelashes. At the optimal length, signal an eye that is well protected, an eye that hasn't lost its lashes to disease, to infection, to the various conditions, both congenital and acquired, that cause alopecia of the lashes. Eyelashes may serve as indicators of an individual's health, since various diseases, both congenital and non-congenital, can influence eyelash length. Short or absent lashes can signal autoimmune conditions, thyroid disorders, inflammatory diseases, chemotherapy, severe stress. Long, full lashes signal the opposite, a body running its follicle cycles without interruption, a system functioning as it was designed to function, an immune and hormonal environment that has not been disrupted by disease. The brain doesn't know it is reading any of this. It doesn't consciously assess follicle health or FGF5 expression levels or antigen to telogen ratios. It produces a feeling, an impression, something about that face, something about those eyes, something that arrives before a word is spoken, before a name is known, before any deliberate evaluation has taken place. And that feeling, which women have been responding to, enhancing, pursuing, and celebrating in themselves and in others since before recorded history, is the product of a gene on chromosome 4, a stop signal for a hair follicle, a molecule that tells a lash when to quit growing, and whose failure to deliver that message produces the feature that 4,000 years of human beings, across every culture and every continent, have called beautiful. In the families from Pakistan, the ones whose eyelashes were so long they caused corneal irritation, who trimmed them regularly, whose children were told at school that their eyes were unusual, there was never a word for what made them different. There was just the experience of seeing the world through eyes that were more framed, more sheltered, more dramatically defined than other people's eyes. The sensation of lashes that swept rather than merely blinked. The parents who trimmed their children's lashes every few weeks, the way most people never have to think about. The gene responsible is ancient. The protein it produces is expressed in the outer root sheath of hair follicles across the entire mammalian kingdom. The mechanism, the stop signal sent at the end of the growth phase, is one of the most conserved biological functions in vertebrate life. And in those Pakistani families, a single mutation in the sequence of that gene, a single amino acid change in the protein it produces, was enough to silence the stop signal entirely, enough to let the lashes grow, enough to produce the feature that humans have been drawing around their eyes with kohl since the time of ancient Egypt, the feature that the brain processes as health and attractiveness before any conscious thought intervenes. The feature that a landmark wind tunnel study in 2015 revealed had been independently optimized to exactly the same length ratio, 1/3 of eye width, by 22 different mammalian lineages over tens of millions of years of separate evolution. The genetics behind long eyelashes is not the story of a beauty trait. It is the story of one of the oldest biological stop signals in the mammalian body, a molecular break that has been calibrating lash length since before the first cat or dog or human being existed, and whose disruption produces, in every species it touches, the longest and most luxurious coat or lash or fleece that species has ever grown. The gene is FGF5. The mechanism is the anagen phase extension. The outcome, when the stop signal fails, is the feature that stands at the intersection of biology and beauty, of medicine and aesthetics, of ancient evolutionary pressure and the mirror on your wall every morning. It is, in the end, a gene that decided when to stop, and the ones who were born without its full function never did.